Hi guys, welcome to my YouTube channel. If you are new here, subscribe to be notified as soon as I upload a new solved past paper video with simplified explanations. Don't forget to like and comment. I'll be glad to answer any doubts. Today we'll be discussing Cambridge IGCSE 0610 Paper 4, Variant 2, from October-November 2022 session. I will be explaining the answers in order to clarify the question and not just answer from the mark scheme. Let's get started. Figure 2.1 is a diagram of the human alimentary canal and associated organs. Table 2.1 shows enzymes, the organs that secrete these enzymes, their substrates and products. Complete Table 2.1. Enzyme. Amylase. Organ that secretes the enzyme, well, that's salivary glands. Remember, its substrate is starch and the product is maltose. Next, organ 3. That's clearly the stomach. The enzymes secreted by the stomach are pepsin/protease. The substrate is protein and the product's amino acids. Okay, so lipase is secreted by the pancreas. Its substrate is lipid and the products are fatty acids and glycerol. Lastly, maltase. The organ for maltase would be the small intestine. From the diagram, it is the organ numbered 5. The substrate for maltase is maltose and the product is glucose. Explain the role of hydrochloric acid in the alimentary canal. One of the primary functions of hydrochloric acid in the stomach is to create an extremely acidic environment. This acidity helps in destroying ingested microorganisms, bacteria, and pathogens that might be present in the food. HCl also contributes to breaking down and denaturing enzymes in harmful bacteria or microorganisms present in food. By altering the structure of these enzymes, HCl renders them inactive and thus prevents their potential harmful effects. The stomach's acidic environment, primarily due to HCl, creates an ideal pH, around pH 1.5 to 3.5, for the activation and function of digestive enzymes, especially pepsin. Pepsin is a digestive enzyme that breaks down proteins into smaller peptides, and it works optimally in an acidic environment. HCl helps create this suitable pH for pepsin and other proteases to carry out effective protein digestion. Inactive pepsinogen, an enzyme produced in the stomach, is converted into its active form, pepsin, by hydrochloric acid. Pepsinogen needs an acidic environment to undergo this transformation. Once activated, pepsin plays a vital role in breaking down proteins into smaller fragments, aiding in digestion. State a function of the region of the alimentary canal labeled 6 in Figure 2.1. From the figure it is evident that the organ numbered 6 is the large intestine. The large intestine performs vital functions in absorbing water, ions, and vitamins while facilitating the fermentation of indigestible food material through the actions of beneficial bacteria contributing significantly to overall digestive and metabolic processes. Describe how food eaten by humans is reduced to smaller pieces and explain how this makes chemical digestion more efficient. You may refer to Figure 2.1 in your answer. Food eaten by humans undergoes a series of processes to be broken down into smaller pieces, which facilitates more efficient chemical digestion. The process begins in the mouth through chewing, mastication, where the food is broken down into smaller fragments by the action of the teeth. This mechanical breakdown increases the food's surface area, making it easier for enzymes to act upon it. As food is chewed, it mixes with saliva secreted by the salivary glands. Saliva contains enzymes like amylase that start breaking down carbohydrates and also aids in the formation of a cohesive mass of partially digested food called a bolus, which is ready for swallowing. The swallowed food bolus enters the stomach, where muscular contractions in the stomach wall churn and mix it with gastric juices. This mechanical action, combined with the acidic environment and enzymes like pepsin, further breaks down the food into a semifluid mixture called chyme. The stomach's muscular contractions, known as peristalsis, help mix and churn the food with digestive enzymes and gastric juices, aiding in the breakdown process. Once the partially digested food, chyme, moves into the small intestine, duodenum, bile salts from the liver and stored in the gallbladder are released into the digestive tract. Bile emulsifies large fat globules into smaller droplets, increasing the surface area of fats. This allows enzymes like lipase to efficiently break down fats into fatty acids and glycerol for absorption. Alright, so here we reach the conclusion of this video. I will be posting the next part shortly. You can visit the entire playlist, and if you have trouble with selective questions, you can watch accordingly. I try to provide detailed but easy to understand explanations so that everyone can follow along and understand the concepts. Thank you for watching. Please like if you found the video helpful and subscribe for more.